endomysium connective tissue in between the cells and around individual cells. Again, the nuclei of the endomysium connective tissue cells. Capillaries run through the endomysium. Capillaries only run through endomysium on all three types. We do see striations. So there's the sarcolemma, the cell membrane. Cut it away a bit to reveal the striations, the right angle bands. You can also see 1, 2, 3 nuclei there. So multiple nuclei, and very, very shallow, actually causing the sarcolemma to bulge out. So not just under the cell membrane, it's actually kind of pushing out the cell membrane a little bit. Long end, we'll come back to that end in a moment. So to give you a sense of nucleus position, as cross-sectional view reveals, nuclei that are just under the sarcolemma. Very, very shallow, very, very superficial in location. Okay? What's wrong with this cross-sectional view? What do they screw up on this model?
They put the nucleus in the center.
See these two? They're wrong. So that one's okay. That one's where you'd expect to find it. These two are too far in. That is an absolute mistake on this model. Go down to the other end. We can see cross-section through the myofibrils. Remember, that's a myofibril over there. So cross-sectional view. Cross-sectional view here. Cross-section through one myofibril there. There's a second myofibril. There's a third myofibril. There's a fourth, a fifth, a sixth, etc. Each myofibril is as long as the cell. It runs the entire internal length of the myofiber, the whole cell. But each myofibril is composed of even smaller structures called myofilaments. So these little dots, each little dot you see, each little teeny tiny dot, each dot in there is a myofilament in cross-section. There are two types, thick and thin. This model makes no effort to differentiate between them. So we're going from myofiber to myofibril to thick and thin myofilaments, from larger to smaller. Okay, and here we have a distal end of a somatic motor neuron. You guys, of course, know about axon collaterals, or collateral axons, the branches of the main axon. So a somatic motor neuron normally has many, many axon collaterals branching off the main axon. Each axon collateral goes to one skeletal myofiber. So this would be an axon collateral coming off the main axon there, going to the motor end plate shown in brown. Remember, it was shown in gray. Where was it now? Oh, yeah, it was shown in gray on that one. Motor end plate of the sarcolemma shown in brown here, where all the nicotinic cholinergic receptors of this cell are located. So the rest of the sarcolemma does not have any acetylcholine receptor. They're all clustered here. So you've got the axon collateral leading to the motor end plate of this skeletal myofiber. There would be the axon terminals containing synaptic vesicles packed with ACH. Action potential, release of ACH onto the nicotinic cholinergic receptors. Open up the gated sodium channels, in comes sodium, depolarization of the skeletal muscle cell. So what the sarcolemma outside the motor end plate has is a bunch of voltage-gated ion channels. And we know what are voltage-gated ion channels associated with? Action potentials, exactly. So if you get a depolarizing local potential, it spreads out to here, opens up the voltage-gated channels, bang, action potential spreading in both directions away from the motor end plate, causing the contraction of the skeletal muscle cell. Any questions?